once we're recording, I'm going to appreciate everybody's patience in advance for any procedural hiccups we have as we launch our first virtual Capstone Festival. Welcome, everybody. For those of you who don't know me, my name is James Lindholm. I'm the chair of the Marine Science Department, and today is our virtual Capstone Festival for fall 2020, the COVID edition. We are today going to have two presentations, both group presentations, from one of my classes, the Marine Science 485, Marine Biogeography of California. So by way of introduction, I would just like to say, because the two groups will describe this in much greater detail, this is a class we created 5 years ago to sample up and down the coast as an annual monitoring project, where the students, the primary deliverable from the class was a report to the state. Now, COVID imposed some restrictions on us, as you know, the students will talk about. But to be perfectly honest, I'm quite excited that we got to do anything at all in the midst of this very weird semester. And I'm excited not only because we got to do something, but the other challenge we face, of course, is that group projects, as we know, are always difficult. And they're difficult in the best of all possible worlds in which you're meeting in a classroom together. They are even more difficult when you're meeting remotely on Zoom. And so I would like to say at the outset that I'm really proud of the group for rallying under the difficult conditions on a number of fronts to complete their two respective projects in a short 16-week period of time. With that, I'm going to start sharing and start the first presentation. So we have two, each are about 20 minutes long. And because they're separate but related, we are going to run the first one, pause, and then run the second one. And then we'll have all the participants, 16 of them, answer questions as a panel. That way that will be our plan. Once I start sharing, if somebody like Monica, who I know would be comfortable doing this, can tell me if she can't see or hear any of the things that we're showing, I'd appreciate it. Otherwise, please stay muted and we will start the presentations. I will be monitoring the chat. So if you want to put questions in the chat as we move along, please do so. And we will try to get to them when the time comes. And we'll discuss that more when we're done with the second presentation. All right, so I'm going to mute.
Yeah, we see your screen sharing. Yeah, we see distribution and abundance.
All right. Good morning, ladies and gentle fish. Welcome to our capstone presentation for MSCI 485. My name is Maxwell Ridlick.
My name is Ashley Belknap.
My name is Jasmine Johnson.
My name is Jacob Toth.
My name is Christina Vance.
My name is Sierra Shirey. So a little bit about the overall importance of biogeography and what it is. So biogeography is the study of the distribution of species and ecosystems in geographic space through geological time. Organisms and biological communities often vary in a regular fashion along geographical gradients, such as latitude, elevation, isolation, and habitat area. So biogeography just looks at where species are from, where they've gone, where they're going, and how they interact with other living organisms. Prior to COVID-19, this course would survey a pretty good chunk of the California coast. So as a lot of you know, California itself is a very long state. It covers 10 degrees of latitude. In the past, Gristle Cove was the most northern site, and La Jolla Cove was the most southern site. And that area the survey area covered a total of 944 kilometers, or also 587 miles. And they monitored species distributions and abundance from environmental conditions like climate change. So in the middle of this state, we have Point Conception, which is considered a transition zone. where species compositions vary from the north and southern regions of the point due to localized currents and oceanographic processes. Previous years have studied both north and south of Point Conception to capture any trends in species distribution. So due to COVID-19, we obviously did not have the luxury of being able to travel up and down the coast, but we are quite fortunate to have the lovely Monterey Peninsula in our backyard. So for the purpose of our study, Point Pinos serves as our Point Conception, and we are looking to see if species distribution changes on the northern and southern regions of this point.
So as Sierra mentioned, this year we wanted to study how different species were distributed specifically around the Monterey Peninsula. We generally studied two different regions, north and south, as you can see in the picture here, but we also wanted to look at any variation within the regions at the individual site level. So for example, in the northern region, was the Maccabee data any different from the Middle Reef data we collected? We've all been to each of these dive sites before, so we had a pretty good idea as to what we should see even before we started collecting data. And we expected to see the biggest difference between the north and the southern regions. But we also expected each site within the region to be just a little different.
So all of us are certified science divers through the American Academy of Underwater Sciences. We were able to get this certification thanks to the incredible scuba program at CSU Monterey Bay. We all took MSCI 380, which is the scientific diving course that is offered prior to taking this course. We were trained by the best to do the best, and due to COVID-19 times, during service intervals and discussions prior to diving and after diving, each of us wore face masks and practiced social distancing. On our way down to the beach and during entry and exit, we kept our masks on our face and regulators in our mouths to reduce our risk of being exposed to the COVID-19 virus. Due to COVID-19, we did have to modify the standard reef check procedure. For those of you who are not familiar with reef check began in 1996 as a simple idea to survey the world's reefs using a standardized method so that surveys could be compared wherever they carried out. Standard protocols survey fish, invertebrates, algae, and include a uniform point contact. We were able to deploy 6 core transects, and a total of 18 data transects were run over the cores, collecting invertebrate, algae, and fish data. Since we had a smaller team of divers than in the previous years for this course, we did not collect uniform point contact data. And all transects that we deployed were 30 meters in length, and they were deployed at varying depths along rocky reefs. At all points during data collection and transect placement, divers hover above the bottom. We often find ourselves in kind of horizontal or like upside down positions, just so that we can accurately count and identify species. And due to this, we are trained to maintain proper buoyancy throughout our entire data collection. We also avoid touching any marine life for both safety and conservation purposes.
Reef Check has us survey 3 different species while we're out in the water: algae, inverts, and fishes. Our algae, as we know in Monterey Bay, is everywhere. With our algae, we did 6 transects that were a meter by meter swath. When doing these surveys, divers were on each side, and we also surveyed 8 species that we saw out there. Our invertebrates were the same. It was another 6 of our 18. Again, another meter by meter swath. This time, we had surveyed for 31 different species. We've counted up to 50 individuals and then recorded their exact distance along the transect. Sometimes our visibility would be bad, and so flashlights were used to get a better image of the invert when we were underwater dealing with bad conditions. Our fishes were our last 6 transect. These were different because they were done 2 meter by 2 meter. Another thing that we wanted was our visibility to be greater than 5 meters to allow us to have the greatest chance to identify the species of fish. We were interested in 35 specific species because these are the ones that Reef Check gives us within our data sheet. Another thing that we wanted to make sure to record was size, because in our terms, fishes smaller than 10 centimeters were considered young of the year.
Our first dive site is called The Wall. It's located just off San Carlos Beach, also known as Breakwater here in the dive community, next to the Coast Guard jetty. This jetty is the inspiration for the name because the long man-made reef looks and feels while you're diving, well, like a wall. As a result, any dive here is pretty straightforward and back, as you can see in our red transects here on the screen. The jetty was created in 1939, and it acts as the southernmost end of the Edward F. Ricketts State Marine Conservation Area. This means that the wall has been around for the better half of a century, and there are also restrictions on fishing or taking from the area while diving. With the time and protection, this site is known to have plenty of fish and wildlife.
Breakwater kelp forest ecosystem has a great diversity of rockfishes. Once juvenile rockfish grow out of their planktonic larvae stage and develop, they return to the kelp forest from the open ocean in large numbers. Young of the year, or yoys, are counted to understand future population numbers. Yoys were the most abundant between fish species at the wall. When you remove the yoys from the data, as seen in graph B, you can clearly see the ranges in abundances from other fish species at breakwater. Black perch was the second most abundant fish species. The most abundant invertebrates were purple urchins, as you can see in graph A. When we remove purple urchins from the data, graph B shows the abundance of the other invertebrates at the wall, including the second most abundant red urchins.
We can see with the algae data that Macrocystis is one of the greater algae in the area, accounting for over 54% of the algae we were able to collect.
Our next site is known as Middle Reef, also at San Carlos Beach in the same Ricketts Conservation Area. This is a natural reef right next to the wall. The reef below is made of rocks and boulders of all different shapes and sizes, split by sandy channels. This provides excellent cover for all different kinds of fish and attachment points for giant kelp that can be seen at the surface. These kelp forests also provide cover for fish that like to swim more toward the middle of the water column, as well as even at the surface. Here, we laid our transects parallel to the shore, as you can see in red, between 16 and 26 feet below the surface.
Senoritas were the most abundant fish present at Middle Reef. It is pictured here below, and senoritas are easy to identify due to their extremely elongated body and prominent black stripe at the caudal fin base. They also are typically observed swimming in loose schools, which may allude to their abundant presence at this site. Some other species present at this site include young of the year, olive and yellowtail rockfish, opali, black and yellow rockfish, pile perch, and grass rockfish. In chart A, we included purple urchins, which are the most abundant species present, and purple urchins were taken out in graph B. And the urchins were taken out to better visualize the species distribution of the remaining organisms counted, and bat stars were the second most abundant species at Middle Reef. The most abundant algae at Middle Reef is giant kelp, which is Macrocystis pyrifera, and this is interesting in regards to the community composition at Middle Reef because the site also happens to be dominated by purple urchins, which feed on the kelp. And the second most abundant algae was feather boa kelp at this site. The next dive site we sampled at was Maccabee Beach. This beach is located in a small protected cove off Cannery Row. It also lies within the Edward F. Ricketts State Marine Conservation Area, and the main entrance to this dive site is from the shoreline in the middle of the cove, which is represented by the pink star in the map below. And this site contains rocky reefs of various shapes and sizes that stick up from sandy flats in the shallow waters less than 5 meters deep. And our core transects were laid parallel to shore with the same methodology of all the other dive sites. And our shallowest transect was in about 6 to 7 meters of water, and the deepest transect was around 8 to 9 meters.
Like Breakwater Wall, Maccabee Beach had yoys as their most abundant fish species. Once we took our yoys out of our graph, it was easier to see how the other species were distributed, and we can see that blue rockfish were the most abundant after our yoys, with several other species of rockfish and perch present at the site. For our invertebrates, our purple urchins dominated this site, and closely followed behind them was the wavy or red turban snail, which had a significant more abundance than the other invertebrates at the site. So it also makes it a little bit harder to see what the distributions of the other invertebrates are, because there are so many more torpid snails.
At Maccabee, giant kelp or Macrocystis pyrifera was the most abundant species, followed by Laminaria farlowi and feather boa.
When comparing algae species abundance between all sites, our results showed that giant kelp is the most abundant at all sites. The site with the greatest overall abundance of giant kelp was at the wall. This is interesting because the wall site also has the most abundant with purple urchins. With the Kruskal Wallace test, there was not a statistically significant difference of algae between sites.
Purple urchins were the most abundant invertebrate species amongst these 3 locations. And as you can see in the chart, the highest counts of urchins were at Middle Reef, and urchins also dominated the individual site analysis as well. This highlights a pattern likely due to the problematic implications of sea urchin herbivory in kelp forest habitats. Monterey-based kelp forests have slowly been phasing into urchin barrens as these species become more dominant in the community composition. However, there was no significant differences between abundance of species presence amongst all dive sites. And off to the left of the chart is our p-value, just supporting that there was no significant differences.
So as Ashley said, we noticed that urchins dominated across all sites, and we kind of just wanted to look and see how urchin abundance varied across our small latitudinal change to see if the regions north of Point Pinos and the regions south of Point Pinos were different from each other. None of these sites are considered urchin barren yet, but surveying and monitoring their populations is crucial for making informed decisions regarding kelp forest conservation.
Between all sites, as you can see in graph A, they're the most abundant between fish species were the yoys. When you look at graph B, when we exclude the yoys, Senoritas were the second most abundant. When we used the Kruskal Wallace test, there was not a significant difference between abundances of fish in the northern region sites.
Biological diversity is the measure of species richness and species evenness. So we ran a Shannon-Weiner diversity index calculation to measure the diversity levels for fish, inverts, and algae at each site. The reason we did this is because in the scientific community, it is well established that ecosystems with higher levels of diversity are often viewed to be as more productive. So here in our graph, we can see that among algae, Maccabee proved to be the most biologically diverse.
Our invertebrate Shannon Diversity Index shows relatively even levels across the sites, even though we had different species richness and evenness for each site. They actually computed to relatively the same diversity level across the northern region, with the wall being slightly more diverse, which may be because there was more evenness there. There was also a higher diversity index for the invertebrates than our fish and our algae, which would indicate that there is more diversity within our invertebrate species than we have within our algae and fish.
So the diversity test for the fish shows a species diversity between each sites. It may look like mid-reef has more diversity, however, we saw higher species diversity at the wall. There may be way more evenness at Maccabee and the mid-reef to explain this kind of graph.
So as you'll recall, the first 3 sites that we've just looked at were in the northern region of the Monterey Peninsula, but now we will be looking at the southern region of our peninsula.
The first site we studied on the southern end of the peninsula was North Monterey, and unfortunately, due to 3 fires raging through the peninsula at the time that we were supposed to collect data, we weren't able to go to the actual site and collect data. So instead, we used virtual reality or VR imagery, which you'll get to hear more about in the next presentation to collect data at the site. However, if we were to go to North Monterey in person, it would have been the first pull-off from Highway 1 between Carmel River Beach and Point Lobos. It's on the northern end of the Point Lobos Marine Reserve, and it's near the mouth of the Monterey Canyon, and it's known for its dense kelp forest and large fish.
South Monastery was also collected using VR data due to the fires previously mentioned, and it is also located between Carmel River Beach and Point Lobos. It will be the second pull-off off of Highway 1, and it has really similar geography to North Monastery, but it recently became an urchin barren, so we expected it to have less diversity and less fish than some of our other sites.
So here's just an overall chart of the distribution of species across all the sites. Here the green represents the fish is present and the red represents that it is not seen there. We can see that South Monastery had the most species seen, which do remember that was done by VR. And Middle Reef had the lowest amount of species seen, which was 7, which was done by our dive team.
So here, looking at our graph, you can see some patterns that might show up. It looks like the southern region with both North and South Monastery shows the highest species richness, or the most species present. We tested the sites using a Crustal-Wallace rank sum test for variance and found that our data actually had no significance. We can't conclude any difference in species richness between the North and South regions of the peninsula, is basically what this test is saying.
In conclusion, we did notice several observable patterns in our data, such as more species seeming to be present in the southern region than compared to the northern region. And we also noticed changing fish communities within the regions themselves. For example, some species were present at Maccabee that we didn't find at Breakwater, and vice versa. However, our statistical tests show no significance in these patterns, surprisingly. In the future, if we were to have more transects at each location and even some additional sample sites, we may be able to normalize our data and run tests that prove these patterns to be significant.
Thank you, everybody. Please hold any questions until after the next presentation. Thank you.
Thank you.
All right, well, thank you for the first group. And I will now queue up the second. What I'd like to point out between presentations, so as they've discussed in the last one, the original dive plan was very different, much more ambitious, and encompassed the entire state. The second challenge we faced is that even with dive operations constrained, the other problem we had is that a majority of the class weren't living in the region, which meant that we had to come up with a different way to engage the students from elsewhere around the state and around the country. And so what the next group is going to talk about is their approach to working at similar locations using similar but somewhat also different approach. So let me share my screen. Okay.
You're sharing. Got it.
Oh, thank you. And I think this is the wrong one. Let me see. Yep. All right. Sorry, 1 second.
Stop sharing your screen.
Got it. Thank you. Oh, there it is. Okay. All right. 1 second. Here we go. Share screen. Computer sound. And go here. All right, here we go for presentation number 2.
Perfect.
Thank you, James, for your wonderful introduction, and good morning, everyone. My name is Isabella, and joining me today is my team, Jose, Krista, Jason, Kathy, Jessica, and Jasmine. And together we worked on our data from our 5 sites, the same ones as previously mentioned by the diving group. The 5 sites span the Monterey Peninsula, and we're excited to share with you that we extracted our data from virtual reality dives. For those of you who may not know, virtual reality is recorded footage that usually spans a 360-degree plane. It puts the viewer in first-person perspective as if they were actually there. And under this semester's circumstances, we had the opportunity to work remotely. We actually got these VR goggles, as you see in the picture, and then also here where we'll put in our phones and play the video from YouTube, and we could look around as if we were actually there. And this is a new and exciting way of collecting data that hasn't been used to its fullest potential yet. And it allowed our research group to collect all of our data virtually. And this was perfect for us because not everybody in our course was a scientific diver. So virtual reality was a new experience for us, and it is definitely something that reaches a broader range of people. We began our project by asking, how does the distribution and abundance of fish species change between sites and between different regions on the Monterey Peninsula? We propose that there will be a change in species composition, density, and abundance between the sites of separate regions. And if you look at this figure, we have 5 sites in total located on the different sides. The navy blue site color represents our northeast sites, and the yellow represents our southwest sites. This coloring will be carried forward for the rest of our presentation.
Video imagery was made possible with the use of these 2 cameras capable of capturing 360 degrees. We have the Abyss 360 shown to your left and the Insta 360 shown to your right. The Abyss 360 was used at the North Monastery and South Monastery dive sites. The Insta 360 was used at Maccabee Beach, the wall at San Carlos Beach, also known as Breakwater, and Middle Reef at San Carlos Beach. Taking note of the housing on each camera as we move on to the next slide. Both cameras were held using a monopole, the exact one shown in the upper right-hand picture. Attached to the end of the monopole in that photo is the Abyss 360 held straight out in front of the diver. Due to the nature of the housing of the Abyss 360, it makes use of 6 cameras that can be stitched together to make a seamless video. The housing on Insta 360, however, slightly interferes with the recording, and in order to counteract that, the Insta 360 was held upwards at a 45-degree angle. Both cameras were about 0.5 to 0.75 meters in front of the diver. The cameras were swam for the entirety of a 30-meter transect at a constant speed, with the diver approximately 1 meter above the seafloor at all times. The photo in the bottom left-hand side of your screen shows an example of one of the transect lines the diver followed. Like Isabella mentioned earlier, the use of VR gave access to those of us who were not scientific divers in the class the ability to do research like this. What that also meant for most of us was that this was going to be our first time IDing fish. Essentially starting from scratch on the screen, you see some of the resources provided to us to be able to familiarize ourselves with fish ID. To the left is a chart of body shape, also noting cross-sections and which fish are associated with each cross-section. And then to the right is a photo of key things identifying a blue rockfish. This is a screen grab from one of our videos. When we do come across a fish, we have a process that we follow. For one, we pause the video and take note of several things. We take note of the timestamp of when we first see the fish. We take note of the quadrant. If you notice, the video has these visible red lines that break the screen into 4 quadrants. This gives us a point of reference to identify where a fish is in association to where we're looking. We take note of the microhabitat. And finally, we take note of what that fish is doing, whether the fish is actively swimming or stationary. And now we'll take a look at a sample video or a sample from one of our videos.
So once we extracted all of our data from the virtual reality footage, we then cleaned our data, organized it, and filtered out all of the unidentifiable fish. We evaluated our data by checking normality, and we ran a few parametric tests, but we discovered that our data was not normal, so we ended up using non-parametric tests. We separated the sites into 2 separate regions and calculated the average species richness per 30-meter unit effort. 30 meters is about the length of 1 transect, and there were 10 transects at each site. We used a Wilcoxon rank sum test to compare between the 2 regions' average species richness per 30-meter unit effort. We used a Kruskal-Wallis rank sum test to detect any significant difference between the average species richness per 30-meter unit effort at any site. We also did this for the average species composition per 30-meter unit effort. In the presence of a significant difference, we used a done post hoc test to distinguish where the difference lies. We then found the highest proportions of individual fish species in each region and focused on those fish to determine any significant differences between sites and between regions in composition of those specific fish.
Hello everyone, I will now be discussing one of our southwest sites in this project. North Monastery is located off Highway 1 between Carmel, River State Beach, and Point Lobos State Park. This brief video to your right shows a small clip collected by the divers at this location. As you can see on your left-hand side, this is our map that shows the transect location at North Monastery. The transect is located approximately 60 meters west from the divers' entrance and extends parallel up the northern coast for approximately 250 meters. The survey area is located on a steep slope with depths ranging from 20 to 44 feet. This video shows the conditions on the day of the survey. Visibility is approximately 15 to 20 meters with moderately calm waters. This is a written sum up of everything I have just mentioned. This location is protected by California's Department of Fish and Wildlife, resulting in high biodiversity. As you can see in our graph, there were many diverse fish in this area. We collected a high level of rockfish as well as blue rockfish and kelp rockfish, and we saw a high level of senoritas and lower levels of perch. I will now be discussing another one of our southwest sites in this project called South Monastery, which is also located off of Highway 1 between Carmel, River State Beach, and Point Lobos State Park. Again, this is a brief video that shows a small clip collected by the divers at this location. This is our GIS map that shows the transect line and the location of the study. The transect is located approximately 70 meters west from the diver's entrance and extends southwest from the coast for approximately 250 meters. The survey area is located on a steep slope with depths ranging from 20 to 66 feet. This video shows the conditions on the day of that survey. Visibility is approximately 15 to 20 meters with moderately calm waters. And again, this is a sum up of everything I have mentioned. This location is relatively similar to North Monastery. It is also protected by California's Department of Fish and Wildlife, resulting in high biodiversity. Again, we collected high levels of rockfish. We saw lower levels of perch and higher levels of school of fish.
For sites categorized as the Northeast regions, our map outlines our breakwater VR transect shown to the map on the left. Our divers have mentioned in their site descriptions, breakwater is located at the end of Cannery Row by the Coast Guard Pier here in Monterey. At this site, you can see the diversity of fish species we saw, including the number of rockfish and blacksmith that inhabit this site on the graph that just popped up. The Middle Reef RV transect is shown to the left with an example of the conditions and sample of the video transect we analyzed for this site on the right. Middle Reef resides within Breakwater Cove about 50 yards away from the wall. And the visualization to the left represents the average count of species seen at this site for 30 meters of effort, with senorita, black surf perch, and pile perch being the most prevalent species at this site. Our last site we analyzed was Maccabee. The Maccabee RV transect is outlined in the map to the left, as well as a sample video of the VR transect we analyzed to the right. Maccabee is located within Cannery Row, so it's a pretty populated area. The visualization, represented as the bar chart, displays the range of species we saw at the site per 30 meters of effort. The abundant species that inhabit this site are blue rockfish, the senorita, and kelp surf perch. The main species we'll be focusing on to compare throughout the sites and regions will be the highest average count species to each site per 30 meters of effort. So these species will be the rockfish, blue rockfish, black rockfish, the blacksmith, olive or yellowtail rockfish, kelp surf perch, and senoritas.
After looking at individual sites, we then wanted to compare between geographical regions. And so as a reminder, on the Northeast Peninsula, we have 3 sites, and that's highlighted in blue. And then on the southwest side of the peninsula, we have 2 sites, and that's highlighted in yellow. And to compare between geographical regions, we want to look at the total count of identified fish. We then want to look at the average species richness per 30 meters of effort. And then finally, the species composition, and that was measured in proportions. We then wanted to compare between individual sites. And we looked at similar things. We looked at the average species richness per 30 meters of effort, and then the species composition, which is count per 30 meters of effort. So we first looked at the total number of identified fish counted, and that's represented on the y-axis. On the x-axis is that geographical region. And we saw a vast majority in the northeast, a total of 858. And if you compare that to the southwest, that's only 219. So in general, we saw a lot more fish in the northeast. We then wanted to compare between the species richness, the 30 meters of effort between regions, and to do so we used the Wilcoxon rank sum test. And as you can see by these error bars highlighted by this red circle, they overlap, indicating that there is no significant difference between the total number of fish species seen between these regions. I'll now let Jessica explain the species composition of the geographical sites.
Thank you, Jason. So this pie chart is showing the different species that we saw through the VR in the Northeast region, which is the blue circle part, as Jason had mentioned. The fish we saw mostly were the blue rockfish as a majority. We have senoritas, which were the second most abundant, followed by the blacksmith, kelp surf perch, and the olive yellowtail rockfish, along with some other type of rockfish that specific species was not identifiable. This pie chart shows the same thing as the previous, but for this is the southwest side, which is the yellowed circle area. Here we see a majority of different types of rockfish that the specific species was not identifiable, but we also saw a majority of blue rockfish and kelp rockfish, along with the senoritas, black rockfish, and of course the olive yellowtail rockfish. Some of these similarities that we can see from both regions is that blue rockfish are the most common, followed by senoritas being the second for the northeast, the blue region, and kelp rockfish for the southwest region, which is yellow.
After comparing between geographical regions, we then wanted to compare between the individual sites themselves. And to do so, we used the Kruskal-Wallace rank sum test. And we found that the average fish species richness per 30 meters of effort was different for at least one of the sites when compared to the others. And then to see which of those sites that was, we used a Dunn post hoc test and found that Middle Reef had significantly lower average fish species richness per 30 meters of effort when compared to Breakwater. It also had lower species richness when compared to Maccabee, and then finally lower species richness when compared to North Monastery. We then wanted to compare between the species composition between these individual sites, and we wanted to highlight the ones that were seen most frequently or in high proportion. And so, as you can see, black rockfish was seen at 4 of the sites, and blue rockfish was seen at the same 4 sites. Kelp rockfish was seen at 5 of the sites, and then olive yellowtail rockfish were only seen at 3 of the sites, but in very high proportions. Kelp surf perch were only seen at 2 of the sites, but again occurred in very high proportions. Senorita were at 4 of the sites, and finally blacksmith were only at 2 of the sites, but again were in very high proportions. And so we took the fish that were most seen or occurred in high proportions, and then wanted to compare between these proportions to see if they were different when compared between sites. And to do so, we used another Kruskal-Wallace test, and we found that there was no significant difference in black rockfish. There was no significant difference between blacksmith, For blue rockfish, there was a significant difference, and we used a done post hoc test to determine that there was a difference in species count per 30 meters of effort between breakwater and South Monastery, which is highlighted by those red circles right there. And there's no significant difference between kelp rockfish. All right, this is the same graph. We're just going to go over the bottom 4 species, and you'll see that there was no significant difference between these 4 bottom species as well. You notice that there was not a lot of statistical difference between these sites. However, this may be due to a variety of reasons. Historically, non-parametric tests like the one we used are typically more conservative, and so they may not find statistical difference. In addition, we had lower sample sizes, and that tends to lead itself to, again, not having very many statistical differences. We think that there may be statistical difference if we did more sampling, because we measured proportions in the geographic regions, as we talked about above, and we did find differences in those proportions. So that might carry over between individual sites. We'll have to do more research to see if that is true.
So to give you guys a break from all the statistics and graphs, here are a few of the fishes we get to see through our VR footage. On the top left-hand corner, circled in red, this is a hidden rockfish that could easily be missed if you were a diver or just swimming along. In the top middle, we have an actual image taken from our VR footage of the blue rockfish at breakwater. Again, this is an image of our most commonly sighted fish in both the northeast and southwest regions. In the bottom left-hand corner, we have an image of our second most spotted fish in the southwest region, which again is the kelp surffish.
From our study, we have learned that Monterey has many small ecosystems. This means that there are a lot of areas where there are good variety of fish and environments that are different from others. So that means that if you are doing a study on the entirety of Monterey, it is a very good idea to take that into consideration. As discussed before, we used a new system of collecting data, which was VR, and there are a couple of advantages that we found from doing so. The main thing is that it's recorded, which means you can share it amongst each other, and if you don't know a fish, or if you're not sure, you can have somebody else look at it and make sure that you have the correct fish, which would help with the accuracy of the study. Another part of it being recorded is that you can rewatch or pause the video, which means you have a lot more time to look at the fish in order to ID it. You can also slow down time as well, which means that if the fish is swimming by too quickly, you can be able to make sure that you see it enough to ID it. However, our study is not perfect. There are ways in order to improve. The main thing is that we were low on data, and the reason why this happened is because we had a small amount of time for our study. We only had 1 semester in order to do this, which was not enough time to collect significant data. We only collected significant data for the blue rockfish, and that was it. If we had more time, then we could have gone to more dive sites and collected more data. We could have also recollected data from sites that had insufficient or altered results. For example, one of our sites had low water quality, which meant that the fish that we saw was not a good example of the general amount of fish in that area. Another way we could have improved was by sharing the video directly. We used YouTube in order to share the videos. However, by doing so, the video quality went down. If we shared the video directly, then the video quality would not go down and we would have a much easier time identifying the fish. However, one advantage to having on YouTube is that you guys can watch them. So you are able to actually see the videos that we recorded for our study. In order to do that, you want to go to California Undersea Imagery Archive, click on Immersive VR Videos, which will take you to this page. And if you scroll down, you'll come across here. And this is our listing of videos. So we have the dive location, description, and the actual link. So if you click any of these, that will take you to one of our videos. And if you scroll down further, they'll take you to our 3-minute videos. I'd like to give a special thanks to Dr. James Lindholm, Dr. Paulatina Salinas Ruiz, Cameron Strickland, Catherine Tensman, Rebecca Richardson, Lauren Wild, Emily Haydes, the divers, and you guys for joining us and watching our capstone presentation. I hope you enjoyed it, and we will go to questions now.
All right, well, thank you very much. And now I guess is the moment where we'd like to ask everybody to unmute and give a round of applause for our two presentations. And then after we've completed our unmuting, let's mute again. Good job, everybody. All right, so if you have any questions, please put them in the chat. I will moderate the chat and ask questions of the group. We're gonna have everybody stay muted except for the members of the class. Please unmute yourself and let's see your video too so we can see you while you're answering questions.
James, your video is off as well.
Yeah, that was intentional, Monica, but I'll bring it back. Thank you. Okay, so first question that I see is, what is the human impact, commercial and sport fishing, pollution, global warming on your research? Does anybody want to take that?
I can take part of that.
Okay.
So human impact as far as fishing and pollution, we do see a lot of pollution, and I believe in the past, correct me if I'm wrong, James, but this class has collected marine debris data.
Yep.
So that's something that previously the intertidal team, instead of having a VR and a diving team, it would be diving and intertidal, and the intertidal team would collect data on the marine debris. So it is something that we look at because it does impact the fish and all species in the ocean. And as far as fishing goes, we see a lot of debris from that, and divers are known to kind of get hooked sometimes, or not like hooked, but tangled in line at the breakwater wall. So that's something that we had to be really mindful of because people were fishing like directly above us.
Thanks, Yara. So the next question is for both groups, and maybe one person from each group can clarify the number of transects per site for each approach.
Yeah, I can take it for the divers at least. So the term transect kind of has a double meaning in this study. What we did was we put down 6 core transects, we're calling them, and those are going to be the physical measuring tapes. And what we did is we put down the physical 30-meter measuring tapes, and then we took our actual data transects, if you will. So we had 6 of the core transects, and then over all of those, we did 3 runs, 1 for fish, 1 for algae, and 1 for invertebrates. So that came out to a total of 18 data transects. But those are just us passing over the physical measuring tapes that we actually laid out.
Thanks, Jacob. Can one of you guys in the VR field that?
Yeah, so for the VR group, we actually measured our transects in video time. So we split up each 30-minute video into 3-minute chunks, and each 3-minute chunk was considered a 30-meter transect.
Yeah, so the transects there were, that you saw the map were just the way we distributed the effort for collection of the video, and then they randomly subsampled after the fact. Thank you, guys. Next question is, can we get a link of this recording? Yes. We'll figure out how to do that. But yes, we will make it available. Certainly all the students will get access to it, and we'll figure out a way to make it public. We have a question. I'm curious about unidentified fishes. Are some species more likely to be difficult to identify, and do some sites have more unidentified species? I guess that's a question for both groups, although the VR probably had a higher proportion of unidentified species. You guys want to handle that?
Yeah, I can take the for the VR team. So the proportion of unidentified species for most of our videos was about 30%, and that mostly has to do with either the distance from the camera or the lighting, where they are in the position of the water column. And there's just some cases where, even though it was recorded, as much as you zoom the video around and pause it and look at it, the lighting just doesn't allow you to see distinguishing marks.
Okay, thank you. Here's a tough one. What's the most fun about this research? Nothing was fun. It was all virtual, and we hate Zoom meetings. Anybody else want to take that one?
I'll take it again for the VR group. I had a fantastic time. We broke up into subgroups to analyze the video, and so we'd get together on a Zoom call, and one person would share the screen, and the other people would then comment, and we'd all work together to identify the fish, and that was a fantastic time. We were all eating, having dinner, and just discussing and looking at fish. It was like being at the Monterey Bay Aquarium, except in actual dive sites. So it was that part was incredibly fun.
Hey, divers.
I can take it again from divers. I mean, at the end of the day, we still got to get out and dive, so there's no complaints on that end. That pretty much sums that up.
All right, thank you. All right, next question. This is a good question. Thanks, Steve. Lots of bar graphs and stats representing lots of good data, collection and analysis, but what does it all mean? What's the take-home message?
I can take this one.
Okay.
I knew that if anybody was going to ask this question, it was going to be Steve. I knew it. So thank you, Steve. I think that it's really interesting because this class does generally survey so much of California. And since we were, we're fortunate to have the peninsula in our backyard, but we aren't able to see kind of as much of the change as previous classes have as far as species distribution goes. But it was cool because we did get to see some changes, and it's interesting to compare like the virtual reality to like the actual diving that we did, because the VR team identified, well, I wouldn't say like identified, but the VR team had a lot more fish counts than we did in the water. So that was something that was really interesting, and just kind of like comparing the two data sets between each other was really cool.
Thanks, Tiara. And as an aside, so one of the things that I think we should clarify is that as the dive team talked about, dive data are collected along a physical transect, and with fish, it's in a 2-meter cube that then you swim through for 30-meter distance. With the VR, they were collecting imagery from imagery in the forward-facing hemisphere of the VR, so it was a much bigger field of view than the diver. And understanding the exchange between that is actually the subject of a new graduate student working with me at Moss Landing to address doing the diver comparison immediately adjacent to the VR, so we can quantify the relative differences in the way they sample the community. So that's forthcoming. Yeah, so this next question, was it harder to get your research because of COVID, and were there limitations? I think the answer to that is probably yes, but you guys want to talk about any limitations that we didn't already bring up?
I can go ahead and talk about some. For the VR, specifically our statistical group, it was really hard just because we didn't have OtterStat, so we weren't working on a web base. So none of us were like working on the same page. We all had the desktop versions, so we were all working like individually. So we were working on like Zoom calls and meetings and stuff, but it was really hard to like get the code to run. I couldn't just like take the keyboard away from like Jason or Isabella and be like, oh, here, it's this. We had to be like, well, try this one. And like, oh, that didn't work. We're like, okay, well, try this. And well, that didn't work. So it was a lot of, I feel like that was a limitation just because we couldn't help each other out as easily as we wanted to.
Not being able to take the keyboard away from your pals.
Yeah, I was like, give me that, let me try.
Next question was, it looks probably like another potential student, maybe a younger student. I noticed that stats was a huge part of both studies. Is stats a major part of the marine science field?
Yes. As much as we wish that it wasn't.
Lots of nodding heads. So it is, and I know we have a couple of our stats professors here on the call, and I think I just want to clarify too, that a class like this, we are consumed with field and analytical stuff, so we're not preordaining a particular test or set of tests the students to do. We ask them to use the things that they've been trained on. So that's what you saw was the best test they could bring to bear on it based on the training to date at CSUMB. So yeah, it's fairly important. All right, so we got a few more questions here. Were VR image taken during the regular dives, or was it taken separate dive just for VR? I can answer that because I did it. It was separate. So for the most of the days, we would stage the divers in 2 separate dives, and in between those dives, the VR would go out. And then on as the group pointed out, when the fires kept us out of the water, air quality improved 3 or 4 days later, enough to us to get into the water at both those locations. So all the VR was collected basically coincident in time with the other data. For either groups, if you had to do this research all over again, would you do anything different? Take a different method? Take a different class? Anybody?
If nobody wants to, I will. But if somebody else wants to, then no.
I don't taking this. So at least something that we were talking about was modality of sharing for the video. I know Kathy brought up that sharing the video through YouTube wasn't exactly perfect. It did lower the quality of our video, so maybe going back to it, we might be able to directly share that video just for quality purposes, and maybe being able to identify more fish. There were also some sites that we couldn't get to just due to life. So we'd probably go back and try to get to those other sites that we had planned. So that's about it.
Thanks, Jose. Another question: what would your advice be for future Capstone students during these COVID times?
Don't panic.
Nice.
Make use of the Google Calendar.
Yeah, that's another good one.
Google Documents and Google Slides for sure.
They're your best friends.
Yeah.
Make sure you communicate with your group members.
Yes.
Yes. Yes.
Discord is a nice tool as well.
Yes. Well, it looks like the questions have slowed down a bit. So I would just like to say thank you again to all the students for all your hard work. And thank you for everybody for joining us this morning. We appreciate your time, and we know everybody's fatigued from Zoom, but we really appreciate your time to spend a little bit more time with us. We wish everybody a happy and safe holidays. And we will be Zooming again next semester. But for those of you who may not be aware, the CSU has announced that next fall we're going to be back in action face-to-face. So we will see how that goes. But for now, signing off. Happy holidays, everybody.
All right, guys. Love you. Bye-bye.
Bye, everyone.
Bye.
Bye, thank you.
Thank you. Bye.
Good to see you, humans.
Bye. Good
to see you
too.
Right.
Thank you, guys.
Nice job, everybody.
Did you see the chat?
Yeah, hang on.
I figured I would have gotten at least a little bit of a facial reaction out of you.
No, I didn't see any of your chat. No, I missed it. Too bad. Must have been too high up.
No, I just sent it like a minute ago. It's at the bottom.
Oh, yeah. No, you can show me, young fellow. Or we could, hang on a second. I'm going to stop recording.